So during my summer internship here at the lab, I studied reversion of hypertrophic growth by DTAC. So the heart is a muscle and it's capable of growing and shrinking based on changes and in response to a variety of stimuli. But unlike regular myocytes, cardiomyocytes are postmeiotic, meaning they don't grow by cell division and instead by a physical change in mass or volume. In recent years, significant strides have been made in deciphering the mechanisms of cardiac hypertrophy, yet little is known about the mechanisms regarding cardiac atrophy. Our understanding the process of cardiac reverse remodeling and discovering ways to inhibit or reverse cardiac hypertrophy may be a way to diminish the widespread syndrome of cardiac failure, which is the #1 cause of deaths worldwide. So why study cardiac atrophy? The aim of my studies was to study the atrophy process within the full range of cardiac remodeling. Clinically, studying atrophy can give us clues into potential therapeutics for hypertrophic cardiomyopathies. Selectively activating targets known to induce atrophy has the potential to reverse hypertrophy in diseased hearts, and although pathological hypertrophy is compensating for an increased load to maintain cardiac output, it has been shown that regression of hypertrophy has no impact on cardiac output. In addition, atrophy itself is a major medical problem. Bed rest, LVAD placement, space flight can cause pathological atrophy of the heart. So most of what we know about myocyte atrophy and hypertrophy comes from studies in skeletal muscle, in growing muscle and hypertrophy muscle. IGF-1, insulin growth factor 1, activates PI3 kinase, which then activates the AKT pathway, upregulating mTOR, and leading to the growth of skeletal muscle by protein synthesis. When the AKT pathway is active, FoxO, the FoxO transcription factor is inactive and phosphorylated, excluding it from the nucleus, and thus deactivating any possible transcription of genes. However, in skeletal muscle atrophy, FoxO is active. With disuse of skeletal muscle, glucocorticoids are released, which in turn inhibit the AKT pathway, and FoxO transcription factors enter the nucleus and activate the transcription of atropines, which mediate autophagy and include MERF1 and atrogen-1, which are E3 ligases, which act to ligate ubiquitin to proteins. Recent publications have shown that MERF1 may not be linked to the reverse remodeling of the heart, although it is still unclear if atrogen-1 has an effect on cardiomyocyte remodeling. In fact, in a publication released this year, an atrogen-1 knockout heart subject to no-load hypertrophy. There are two types of catabolic cellular processes within atrophy. Firstly, there's proteasomal degradation of cells. During this process, proteins are targeted by E3 ligases, such as MRF1 and atrogen-1, which ligate ubiquitin to the target protein, and which then the ubiquitinated protein will then be degraded by peptidases in a proteasome. Secondly, there is autophagy, which literally means to eat oneself. It's a term of for the degradation of cytoplasmic components within lysosomes. It is mediated by autophagosomes, and as autophagosomes engulf a portion of the cytoplasm, it is generally a more non-selective process as opposed to the ubiquitin proteasome system. So my hypothesis was that FoxO transcription factors are necessary for the reverse remodeling of cardiac muscle. So before moving on, I'll give a little background into what I was studying. FoxO transcription factors are a subgroup of the fork-head family of transcription factors and have been shown to be at the interface of crucial cellular processes such as proliferation, differentiation, metabolism, and cell cycle regulation. There are two major subtypes of FoxO transcription factors known as FoxO1 and FoxO3 that have been shown to be expressed in cardiomyocytes. These and that have been shown to play a crucial role in skeletal muscle atrophy. So to simulate cardiac stress and load, we subjected mice to severe transverse aortic constriction, an STAC, for 1 week to induce hypertrophy, after which we removed the constriction, the DTAC, and observed regression of cardiac hypertrophy initially caused by the original STAC. I'd like to thank Herman for, because he conducted all the surgeries on the mice. So we examined regression of hypertrophy by heart weight to body weight ratios. Although in the figure the data is not significant, it's clear that there was a regression of the heart after removal of the TAC.
So that's 20, is that 7, one in the middle is 7 days, one in the right is 14 days?
Yeah.
Yes. So
We can go back for a second, just so we're, so the reason it's not significant is we only had a couple of STACs, most of them. So I think once the numbers are higher in STAC, I think the model works. It should be higher. More numbers.
We're gonna do 1 week, so
No, I think that we just only had like 3 mice for the STAC. So there's a huge error bar, whereas at DTAC we had about 8. Yeah. And to be fair, those sham are from a different study, but sham.
okay, so
They're same gene type though.
to knock out the FoxO protein, we crossed a mouse carrying the Merck-Creamer gene driven by an alpha MHC promoter, meaning that it will only be expressed in the heart, with a mouse that carries a FoxO gene with LOXP sites inserted into it. Animals now positive for floxed FoxO and alpha MHC pre. When injected with tamoxifen, the tamoxifen binds the Merck-Creamer, translocates to the nucleus and recombines at LOXP sites, which removes the FoxO3. Thus, the FoxO3 is removed from the cardiomyocyte genome, thus the FoxO3 data suggests that FoxO3 transcription factor is not required in the reverse remodeling of the heart. Knockout hearts exhibited the same growth and atrophy as wild-type hearts. Although it does appear that FoxO3 is not responsible, FoxO1 transcription factor may compensate for the loss of FoxO3, but further testing needs to be done.
So you injected the tamoxifen and waited how long before you did all of it?
2
2 weeks? And everybody was fine by then? The heart was-
Yeah, I mean, Herman did some echoes.
By 2 weeks, you were okay?
Yeah, I think most of the time.
Is FoxO3 changed during by tech? I know FoxO1 is changed, right?
Consistent. In the 3 different heart, 2 you can see also 3 you can not that much. In 1 heart you see it.
It goes up.
But the question is, does the FoxO3 still hypertrophies? Yeah, that was the question. The knockout still hypertrophies.
No, we
You
wild
know,
type.
The wild type, do you see change?
Where
in
you see
FoxO3?
change like that.
Yeah.
You know what, we had problems with the FoxO-Westerns and FoxO-Western, so we never really get that data. And that was one of the things we wanted to see, whether FoxO was changing, but you know, we never really got that data. FoxO, no relation.
And you know it recombined that FoxO here knocked down?
Not 100% again, because we used the FoxO3 antibody. I mean, we saw the problems with the total FoxO antibody, you know, we got a signal. There didn't seem to be much decrease, but, you know, we're looking in the background of FoxO. So you really need to get the FoxO3 antibody working. We weren't able to get it to work.
So next we wanted to look at molecular changes. So we first looked at the catabolic pathway of autophagy by examining levels of LC32. LC32 is just a faster migrating variant of LC3 that indicates lipidation and formation of autophagomes. It's here we have it quantitated as a ratio of LC32 to GAP-DH, and it appears that steady-state levels of LC32 decreased after DTAC. Next, we looked at a direct target of FoxO atrogen. The atrogen protein levels increased during DTAC, and they seem to be slightly less than the FoxO3 knockout animals.
Where's your computer?
There, that's got the age of 37, and that wasn't in there.
Don't make fun of his GAP-DH, man.
No, we are having trouble with the gate.
Yeah.
What are you talking about? It's the most reliable way.
Not if you only do the incubation for an hour.
No.
Even 30 minutes.
We always do 30 minutes.
Okay. Yeah. 30 minutes.
You guys are GAP-DH snobs.
You probably got a better. Which antibody do you use? The one from
The one free.
from Santa Cruz?
No, never.
That's why. This is the Santa Cruz. This is the Santa Cruz antibody.
Which one do you?
The pizza gal.
Because
Pizza
I,
gal. There's a weird name company.
Okay, well that's probably
The
why.
mouse?
I couldn't find that one. I
Mouse
only
one?
found the Santa Claus
I
one.
don't remember the name, but it was really weird.
All right, well, we can,
oh, we can use that.
30 minutes.
Because,
I think people who have a problem are using the Santa Claus antibody.
no, in the lab, we had a mouse GAP-DH antibody that worked good, but we are out of that antibody. So we went back to the rabbit one, the Santa Cruz one. Study.
Yeah, Santa Cruz one's terrible.
Will you share your fancy number?
I'll charge for it.
This
Job
is
has,
the-
Job has
I'll make
its
a
own
fortune on it.
inventory that you can.
This is the, yeah, this
Nobody
is
can get
this
in.
is the Santa Claus antibody.
Yeah. Someone stole it like a couple weeks ago. Someone stole this whole one.
Good.
Yeah,
Who
we
is
have,
it?
now we have a new one.
See?
Careful. Secret stock, all right. We'll have to-
Job mislabeled the queue so that nobody can. Oh my God.
That's how people look.
That's it.
So we looked at the catabolic pathways, but we also looked at the anabolic pathways. Inhibition of AKT activates FoxO and should lower the activity of mTOR. So we looked at a downstream target of mTOR, phospho-S6. We expected that phospho-S6 should be up in STAT during hypertrophy and down during atrophy. But in our hands, the S6 phosphorylation was all over the place, and we're not sure of what it means, and it needs to be more carefully looked at.
What time of the day did you?
Yeah, no, that's the thing.
I'm serious, it's really important.
I bet you that's it, probably has time.
So there's been a time for it,
Yeah.
and there's a time of the day that even if they're attacked, S6 is dead. And that's usually around the middle of the day.
Really?
You should.
Yeah, you're right, and I know that after I looked at it, that's exactly what I figured happened. And that was a time of day issue, because,
So I did this mistake too.
you know, yeah.
And so the STAC-induced response is swamped by the circadian.
Yeah, you have to make sure everything is sacrificed at the same time of day, and these certainly, I know they weren't.
Well, the first time I see the second one, let's say 12 o'clock, and then on the fourth is probably almost 12:30 or something.
So, in conclusion, the release of ligature after STAC results in a reversion of hypertrophic growth. This atrophy response correlates with an increase in androgen 1, loss of FoxO